Good morning. Let's discuss on ammonia metabolism and its toxicity. So, in the body, the major sources of ammonia are the transamination and deamination reaction, degradation of purines and pyrimidine, biogenic amines by the action of intestinal bacteria. You can see it here. So, glutamic acid reaction, glutamine acid via the transamination reaction it converts up into the alpha ketoglutarate, releases ammonia via the action of glutamate dehydrogenase, then deamination reaction, then degradation of amino sugars, then pyrimidines and purines degradation, then putrefaction caused in the intestine by the intestinal bacteria, then glutamine and asparagine can also be broken up into glutamate and aspartate, and urea and ammonia is released. So these are adding to the pool of ammonia in the body. So ammonia can be utilized for production of glutamine via concerned synthetase enzyme. Then it can also synthesize asparagine right from aspartate, and then urea synthesis. Final disposal of ammonia happens in the form of urea because it is the least toxic form of nitrogen. While ammonia is very toxic, it is to be immediately scavenged out if the level raises in the body. So major sources of ammonia have already discussed. Transportation of ammonia happens in the form of glutamine. It is the major temporary storage as well as the transporting form of ammonia. Then disposal of ammonia in the form of urea in the liver by the reaction of urea cycle that we will discuss later on. Then disposal of carbon skeleton of amino acids after the removal of alpha amino group. So the carbon skeleton of 20 amino acid is converged to form only 7 major products. So these are the 7 major products which are all related. These are all basically intermediates of Krebs cycle: pyruvate, acetyl coenzyme A, acetoacetyl coenzyme A, alpha ketoglutarate, succinyl coenzyme A, fumarate, and oxaloacid. These are all intermediates of Krebs cycle, and these are being synthesized from the carbon skeleton of amino acid, 20 amino acids. So from here, the carbon skeleton are diverted to gluconeogenesis or ketogenesis, or they can completely be oxidized to carbon dioxide and water. So these can be the fate of these intermediates. In general, for energy production, they all of them converted to carbon dioxide and water. So in condition of fasting, so once there is a lack of glucose, so these intermediates may convert or divert to the synthesis of glucose via gluconeogenesis pathway, and they can also be used for synthesis of ketone bodies via formation via acetyl coenzyme A and acetoacetyl coenzyme A. So this is the regarding the transportation. So from extrahepatic tissues and muscles, the free ammonia is transported to the liver in the form of in two principal form. One is glutamine and the alanine respectively. Like from extrahepatic tissues, it is being collected and transported in the form of glutamine, while from the muscles in the form of alanine. So you can see the reactions. So glutamate, from the extrahepatic sites, it picks up the excessive ammonia and converts up into the glutamine in the presence of enzyme glutamine synthetase. And this glutamine in the liver, this glutamine is broken up into the glutamate and ammonia via the glutaminase enzyme. For final disposal happens in the liver only. The glutamine so formed is a neutral, non-toxic compound which can readily pass through the cell membrane, whereas glutamate cannot. So it is always the preferred one as far as transportation all through the body is concerned. Glutamine is the major transport and temporary storage form of ammonia from brain and intestine as well to the liver. So we can see the transportation of ammonia from the muscles to the liver via the glucose alanine cycle, and what is the role of alanine. So you can see this is the how the glucose alanine cycles runs and how it contributes to the transportation of ammonia. So we can see it here in the muscles there, whenever there is a muscle a protein degradation, turnover is there, amino acids, especially the branch in amino acids are released. These amino acids release the ammonia nitro or amine group via transamination and transfer of these amino group to the keto acid, formation of glutamate, and this glutamate exchange its amino group to pyruvate, and pyruvate turns to alanine, and this via that alanine amino transferase enzyme, then release of alpha ketoglutarate. And this pyruvate comes from where? Whenever there is a muscle activity, glucose is utilized via glycolysis, converts up into the pyruvate. This alanine via blood, it goes to the liver. Here in the liver, again through transamination reaction via alpha ketoglutarate, this alanine is converted to pyruvate, and while alpha ketoglutarate turn into glutamate, which releases ammonia via glutamate dehydrogenase enzyme, and it converts up into the urea. While pyruvate converts to glucose via gluconeogenesis pathway. So this is how we have converted pyruvate into the glucose, and glucose via blood it goes to the muscles. Muscle again it converts up into the pyruvate, then pyruvate to alanine, alanine goes to the liver via blood, and again pyruvate glucose. So this cycle goes on, and this is actually the glucose alanine cycle. So this is how the scavenging of amino group, indirectly the amino group is scavenged out, and via alanine it's loaded in the form of alanine, and it goes to the liver, and here liver it's being exchanged with the alpha ketoglutarate. This amino group is now funneled into the glutamate, and from glutamate ammonia is released via glutamate dehydrogenation or deamination reaction is there, and this ammonia which is being released is used for the synthesis of urea. So move on to the toxicity of ammonia. As we know, the ammonia normal concentration in the blood remains 15 to 45 micrograms per dl. So once it concentration raises beyond the range, it leads to toxicity, and the higher faculties like brains are mostly affected. So what are the changes happen in course of toxicity? And the first is the formation of ammonium ion. Severe ammonium ion leads to the alkalosis, so change in pH, and then depletion of alpha ketoglutarate to and ATP is there, because alpha ketoglutarate is going to pick the excessive ammonia and it converts up into the glutamate. This is the reaction. Alpha ketoglutarate is converting up into the this alpha ketoglutarate is converting here in this reaction to glutamate, and glutamate is converting to glutamine. So what is happening in course of this reaction, especially in the brain? So what happens here? There is a lack of alpha ketoglutarate. It is such an important intermediate of Krebs cycle. So Krebs cycle will slows down and leading to the less production of ATP. Secondarily, the formation of glutamine and GABA, and because of formation of glutamine, what happens? Because glutamate is diverted to the synthesis of glutamine, so it is no more available to synthesize GABA. Here we can see the glutamate is required for synthesis of GABA via the glutamate decarboxylase reaction. So the GABA is an important neuroinhibitory transmitter, so there is lack of this GABA. And then secondarily, the glutamine leads to so many biochemical changes uh, in the brain. Like it leads to the swelling of astrocytes due to the osmotic imbalances caused by glutamine, and this leads to encephalopathy. And then secondarily, it increased tryptophan level in the brain, because via the anti-port mechanism the glutamine goes out of the brain, and it leads to, in exchange, it leads to the entry of tryptophan. Excessive tryptophan goes into the brain, and here is excessive formation of serotonin is derived from the tryptophan. So increase level of this serotonin will lead to excitatory responses, then increased permeability of neuronal membrane to potassium and chlorides on ions. So these are the events caused due to the toxicity of ammonia.